The Center for Integrative Environmental Health Sciences and Department of Pharmacology and Toxicology welcomes you to our Environmental Health Science Series seminar. It is my great honor to introduce today's speaker, Dr. Lu Kai. Dr. Kai received his medical degree in general medicine, a master's degree in radiation genetics, and a Ph.D. degree in radiation biology oncology, all from Norman Bethune University of Medical Sciences in Chongqing, China. After completing his degrees, Dr. Kai moved to Montreal, Canada, where he completed a postdoctoral fellowship at McGill University. He then moved to the University of Louisville, where he was appointed as an assistant professor in the Department of Medicine. Dr. Kai now holds many positions here at the University of Louisville as a tenured professor in the departments of pediatrics, radiation oncology, and pharmacology and toxicology. He has also served as a visiting professor at multiple universities. Along with these faculty positions, he is also the director of the Pediatric Research Institute at the University of Louisville School of Medicine. Dr. Kai has also held several positions in national societies, mostly recently serving as president of OVSOT. He has served in many editorial positions and is currently the editor-in-chief for the Journal of Microbial and Biochemical Technology, the World Journal of Diabetes, and the Journal of Toxicology and Risk Assessment. Dr. Kai has trained more than 40 research associates and postdoctoral fellows and mentored 15 graduate students. He has authored or co-authored more than 260 peer-reviewed original research articles, 10 in the last year, 80 peer-reviewed reviews, and 10 book chapters. He has received several honors for his work, including a Medal Specialty Section Career Achievement Award and a Distinguished Chinese Toxicologist Award from the Society of Toxicology. Dr. Kai's projects have been continuously supported by the American Diabetes Association, the American Heart Association, the Juvenile Diabetes Research Foundation, and the Department of Defense and the NIH. He's also serves as a co-investigator on several grants with distinguished U of L faculty. Dr. Kai's scientific work focuses on investigating the etiologies of environmental toxicity, obesity, and diabetes. Today, he will be presenting his work entitled Diabetic Complications, the Role of Zinc and Metallothionein.
Good morning, everyone, and thank you very much for your coming, and also as the people online. And so it's a great pleasure for me and introduce some work in my lab. And I think the first seminar for the MOT rig and a seminar is 2 years ago. At the moment, I overview my research regarding the cadmium and study. So today I want to introduce some part of my major focus on diabetic complication research, and specifically emphasize how important zinc and with metallothionein protect the diabetic complication. And I want to divide the full part to introduce my talk and zinc and metallothionein and how zinc prevent diabetes, and use the diabetic cardiomyopathy as the model. And the last part, and give you some from the literature and the clinical evidence to support or the link of my research. So before that, and I want to ask a question, because that's very special. And is there anyone know when is the Diabetes Aware Month? Look like no, but it's November. Is the day, and I want to present. So November actually is the month, and ADA and several years ago tried to define that, but not really recognized until 80s. So now is well accepted, and NIDDK and the GDRF and the ADA, and they have the blue circle as the one a very universal symbol for diabetes. So that is something I learned recently. Another thing is one is the World Diabetes Day. No one? Try? Okay. Also November, yeah. November 14, and that is a World Diabetes Day, and created in 1991 by IDF and WHO. And why they pick up the November 14? Because that is Dr. Bunting, and he discovered Banting's birthday, and he discovered insulin in 1922, and worked with Dr. Charles Best. So I want to give you a small story. I visit and Dr. Jin, and he know I went to that moment, went to Montreal for a mental meeting. And after that, he invited me to go to Toronto, have a short visit with his lab. He worked on the FGF21. And in this building, and this building, you can see, this is a big complex building. They have a different apartment. And one part, they have kind of the very emphasized the insulin contribution for the world. And they have one window and exhibition, and they have a lot of document regarding Dr. Banting. And this table attracted my attention because that table is made by a tree grown up in the front of the Toronto University campus. And they use that table to discover the insulin. So, and this is the ring is one of the four and made by the metal from the aircraft. That aircraft killed Dr. Banting. And after he discovered the insulin that is 100 years ago, and 19 years later, and he died, so that is too bad. So this one ring keep there as a memory. So I will show what is the outcome after I visit them, and later at the end of my talk. So now I can go back to the talk about why I'm interested in zinc. How important zinc is very important. That is an essential metal in our body, and it's very important for many cellular events, and that is required for more than 300 enzymes, transcription factors. That is required for DNA repair and cell proliferation and wound healing, and also is maintain our normal immunofunctions. And zinc is also required for baby growing, so that is tissue growth. And that is also required for maintain our normal taste and smell. So when you got COVID-19, you lose that because of zinc deficiency. Why zinc can help for the COVID-19? But more important for me and interesting is the importance of zinc for insulin, because the zinc insulin structure in need of zinc makes the normal structure, and also is required for the insulin secretion from the beta cells to release to the blood, and also is required for the insulin signaling pathway. And the feature of the zinc in our body is also unique. We do not have a free zinc. We have to daily or routine take the zinc to maintain our balance. Why? We have to understand how the balance in our body, how regulate. So in our body, they have a two group protein. One group is called ZIP, from 1 to 14. So ZIP and try to make zinc and to go to the cytosol. And no matter it's out or some organelle. And another group is the ZIN, ZNTN. So that is make the zinc out from cytosol. So you can see ZIP from outside cells makes the go to cytosol. But if they have too much or binding, they make the zinc go to the nuclear, go to the organelle, and some small component. And so this makes it out or go to the other component. So they do not have a free zinc, but where the free zinc come from? So that from MT. We call what is the MT? Metallothionein. Metallothionein is a small peptide, only 63 or 67 amino acid, but 1/3 is a cysteine, strong free radical scavenger capacity. And then normally the metallothionein binding zinc can a peptide can binding 7 zinc. So when you have required the MT just donor and zinc to the protein they need, or the storage or the secreted. So that feature makes the MT have a very strong antioxidant function and also play very important for mitral homeostasis. Why I was a postdoc, a research associate, come to Louisville, like Mark and just introduced me. And I work with Dr. James Kang and start to think about the diabetic complication. So I learned and diabetes causes the superoxide, and the superoxide generate different kinds of free radicals. And if we only use the SOD, or catalyst as an antioxidant, or with vitamin E to prevent diabetic complication, but doesn't work because they only work one part in the puzzle. So we need some good antioxidant can work more and probably better. So our hypothesis is that probably metallothionein is the good candidate to protect the heart or some other organ from diabetes through the antioxidant functions. So now I want to move to the next part. So how zinc protect the heart and from the diabetes. And I just mentioned, and other antioxidant, SOD or catalyst, they only work on the one part. But we try to look for something work more. And 20 years ago, and I know MT can work all this part, but we don't know there. So collaborate with Dr. John Klein or and Y. James Kang and by myself, we solved the puzzle. We found the metallothionein can inhibit peroxynitrate-induced DNA damage and protein damage in the in vitro and in the cell-free system. So we solved the puzzle. So we know metallothionein can work all the different puzzle part. So and we use the transgenic mice, specifically overexpress metallothionein in the myocytes. So that means only in the heart have MT higher. So we found superoxide high in the wild type, but not in the MT overexpressed in the heart. So that protects heart from diabetes-induced damage. So through the several projects, sorry about that. Through several projects, and we found, and MT really protect and diabetes causes the intracellular event that is AGN2 or diabetes through the receptor TRNOX pathway generate superoxide, and peroxynitrate cause the 3-nitrotyrosine formation cause apoptotic cell death. Because heart myocytes not regenerate. If die, they are turn on some other infect. So that is a cause of inflammation, cardiac cells hypertrophy, and generate some fibrosis response. So lately, and were dysfunctions. We found metallothionein work in the top part and can early prevent the RS generation, and also can scavenge the RS to protect and diabetes caused RS and oxidative stress to protect the cell death, to protect the cardiac dysfunction. So after we understand overexpressed MT in the heart can protect from diabetes, and one postdoc, Dr. Gu, and asked probably knockout mice will be more sensitive. So we use the model and the high-fat diet plus STZ induced the kind of type 2 diabetes. And this is the model showing you the MT knockout mice really did not show MT and the quantity, and you can see that. And we want to look for the cardiac damage, like hypertrophy as one marker. We have a multiple measurement, but I bring here only hypertrophy. You can see in the diabetes group, the myocytes become large. So this is the staining for the membrane. So in the circles that indicated the cell size. And you can see in the wild type, diabetes caused that much increase compared to control, but in the knockout mice, further increased. So other like AMP or beta and MHC, and that is one Western or RA method to measure the the expression for the protein. You can see wild type increase and knockout further increased. So that confirms the MT protect in the heart from the diabetes in the different kind of animal models. Does that have some clinical relevance? So we collaborate with my colleague from China, and they use Chinese population to screening to see if the population with MT gene mutations, they have a high risk for diabetes, high risk for cardiovascular disease. And that is make me very happy. We have some clinical relevance. But furthermore, and see, meanwhile, when we published that in 2008, May, and you can see another publication, 2008, May, in the same year and the same month, published the same things. They use Italian peoples, and they also found metallothionein gene mutation, high risk diabetes, high risk cardiovascular disease. So really relevant for clinical. So now I want to understand mechanism. Is that zinc and can induce MT protect heart or not? So Dr. Wang and working in my group long time ago, and use zinc to induce MT in the heart, try to look for protect the heart from diabetes or not. And we use the STZ model, induced type 1 diabetes, and we give the zinc treatment and different time to look for its cardiac MT increase or not. So we use the staining, we use Western, we really see the MT in the heart increased, but not other antioxidant. And when we look for the different kind of measure to indicate the damage, you can see the staining structure change, diabetes group significantly increased in this part. And the EM increased, and the collagen formation increased, and the CTGF and increased, and the zinc treatment really protect. So we plus the in vitro study, we confirm that zinc induced MT protect the heart and plays a major role, and better than only zinc if we knock down SRA, knock down the MT. So that is the mechanism. So after we published that in the circulation in 2006, and another study, clinical study, probably they read our paper. I just guess, I hope. And they published one from the humans, type 2 diabetic patients. So they measure their blood zinc. They cannot measure heart MT, but they measure the zinc in the blood. You can see the lower one, they have a high cardiovascular complication. The higher zinc group have a lower. So then we confirm our animal preclinical study, the zinc can induce MT protect organ from diabetes. So they make the conclusion in type 2 diabetes, low serum zinc is an independent risk for cardiovascular complications. So under the Dr. Gu, don't want to stop, continue ask the good question, and we want to use a type 2 diabetes DBDB mice, because before we use the zinc-induced diabetes models, you can see in this is a DBDB, normal zinc, and the cardiac function significantly increased. So that is like a dysfunction. But one has zinc treatment, and that is a reverse, prevent that happened. So we also use like high-fat diet, induced like a young mice from the 3 weeks old, give 6 months high-fat diet feeding, and that causes like a type 2 diabetes in early stage and causes the cardiac hypertrophy. We found zinc deficiency worse that, and zinc supplementation can prevent that. And the further study, we found that is the prevention is through the metallothionein dependent, and the BCL10 and CAR9 P38 kinase pathway. So that is really shown the zinc independent need a dependent MT to protect the heart. So that is through some like antioxidant function, and zinc play other function, like I mentioned, the zinc insulin signal pathway. So we look for that, and in 2009, one postdoc and start to ask this, and we found the MT not only protect the antioxidant and the oxidative stress and like inflammation, but can direct work on the GSK surrogate pathway, can make that activation and to inactivate phosphorylation increased. So after that, we further want to understand more and for AKT pathway. So Dr. Gu and work on that part. You see the wild type diabetes reduce phosphorylation AKT and total AKT phosphorylation. and MT transgenic mice prevent that inhibition. When we look for the different isoform, and we found AKT1 not that much decreased, but you can see AKT2 significantly decreased. And the following the downstream and the glucose metabolism, GSK and HK2 and decreased, and PGC1 alpha, but TRB3 increased. So what we found diabetes like specifically inhibited the AKT2 and the associated downstream pathways. And MT can prevent that part. And we found that is MT and zinc is through prevent oxidative stress, inhibit TRB3, and inhibit TRB3 inhibition of the AKT2. So probably you will ask, what is the TRB3? TRB3 is the one protein in response of ER stress. And diabetes condition, the TRB3 and is increased. And if we shut down and TRB3 really can prevent and improve diabetes-induced cardiomyopathy. And how they work, and because the TRB3 specifically binding AKT2, one TRB3 binding AKT2 that make AKT2 degrade. So that is a specific remove AKT2 in the heart, not AKT1. Okay, so we found AKT2 really important for our research. So when we look for literature, how important, and we found that and 20 years ago, they already understand if mice have a AKT2 and deficiency, and that is shown typical type 2 diabetes models, phenotype. And later they found in the human, same thing. If they have a AKT2 gene and mutation, they shown like a phenotype of type 2 diabetes. So really important. So we learned that. So we ask probably zinc MT protect the heart from diabetes is pivotally dependent on the AKT2. So we use our model cardiac specific overexpress MT model to crossbreeding with the whole body AKT2 deficiency mice. So what we can get? So we should get one mice, one line that have a whole body AKT2 deletion. Only in the heart have MT. So we will see what happened. So that 6 months should be typical type 2 diabetes with complication because like we want to look for that, what happened. We have a whole bunch of data, so I don't want to give you detailed results, but give you summarize. So this is the signaling pathway insulin turn on both AKT1, AKT2, that AKT2 plays a major role and to turn on the glucose metabolism, at least in the heart. When we have AKT knockdown, and the animal still survive, but they are increase more AKT1, so that turn on a little bit the glucose metabolism pathways. That's why they are not die, but more sensitive, and the spontaneous development cardiac hypertrophy, fibrosis, dysfunction, and because the downstream glucose mechanism is shut down. So when this mice, we give the MT overexpress in the heart, and you can see that adapt and increase AKT1 is gone. So what happened in this part? And we don't know. And but we found the glucose metabolism pathway revised, and the complication significantly reduced. So we are working on what is the link, how MT can make the glucose metabolism pathway can go back to normal without AKT2. Without the AKT2 plays an essential role, we lose the protection, but not. So this sense, sometimes you will have some unexpected data. So we continue to ask, is that zinc can help? So, and one student work on that, and we use a AK2 knockout mice and give the zinc supplementation, and you can see the glucose level. So this is before we gave the zinc, that already typical type 2 diabetic model, glucose significantly increased compared to control. And if without zinc treatment, that keep on the high level. But if we have zinc treatment, you can see that gradually make the glucose level go to down. So the insulin resistant and got like improved. And for the cardiac function, you can see AKT2, they have a significant dysfunction, but not like in the AKT2 with zinc and supplement. And this is some measurement, same thing, AKT2 kind of diabetes and shown all the increased and but not in the zinc treatment. And this is like AKT and pathway, and you can see that kind of is revised. But and later we found that revised is metallothionein dependent, because if we do not have metallothionein and the expression, also like a revised is going down. So they lose the revised. So that means the zinc MT can rescue AKT2 depend each other. How they work together to rescue, we don't know. That is our next part. So basically our study and shown that diabetes causes oxidative stress pathway, and zinc and MT based on the cysteine pathway, so they can significantly protect that. And we know AKT2 plays a very important role in the glucose metabolism, and diabetes inhibits this pathway, and through the TRP3, and MT can prevent that, and zinc can stimulate glucose metabolism. And actually, they have a more study coming, and just not from my group alone, and they found zinc is kind of and involved the insulin signaling pathway. So that is our group. We work on diabetic complication. We work on heart and nephropathy and wound healing and other organ damage. And we use MT and zinc. And we recently found NERV2 is like an upstream mediator, can turn on the MT, but the zinc can stimulate both heart. And we use some like sulforaphane as an activator, and also use some the drug clinical used with two purposes to use, and through the signal pathway. So that's our work. But I want to give you some evidence. How about the clinical trial, clinical data? Not from my group. I don't have a patient. But you can see this is the one and like a systemic review and meta-analysis, and they based on the case report, a human study from like more than 3,000 cases, and based on their selection and step by step, finally they found only 27 cases is like qualify for their analysis. They have a very detailed table that, and I'll give you a summarize here. So this divide to like a dose group, lower than 25 per day and higher 25. And the time is lower 3 months, and one is like over 3 months. And you can see fasting glucose and levels. So lower 25 milligram, you can see significantly reduce and your glucose levels. And the short time compared to longer time is better. And for the hemoglobin, HbA1c, so that is a glycated hemoglobin. So that is actually is a stable marker for diabetes. So you can see that all the treatment and shown the good and the results, especially lower than this and longer time. And this is like one insulin resistant marker. You can see zinc really significantly improved. But for this like a measurement, somehow is longer is not good. And for the like TG and lipid profile, and at least not that significant, but still better than not used. So this is several other like a case report. I don't know what time right now. Okay, so we have so this is a couple like a case report. You can see they have like 70 patients, and they gave like a 35 patients like 2 group, and then they gave it 50 milligram zinc per day and for 8 weeks, and they shown the good and the results. And this even like we know that a patient with that beta thalassemia that have a whole body have some hemoglobin and the high iron and like situation, so that easily cause the diabetes. So even that's the case, they also give the zinc and also shown the very good protection for the complication, change the profile. So why zinc that really help? Because diabetes somehow affect zinc absorption and increase absorption limitation from our body, because probably high glucose makes the kidney and not very best to resorb the zinc. So that diabetes easily causes zinc deficiency. When we give zinc, and that is can upregulate your body's zinc, can turn metallothionein, can turn modify the zinc transporter, so comprehensively will prevent diabetic complications. And so that's why, and I already work on that field for several years, keep cool, so we can use zinc to fight diabetes, to fight diabetic complications in the different journal at a different time period. And what outcome? And I want to give you one example. Here I have one online, and you can see work and expertise you can find, right? So if I type the diabetic cardiomyopathy, guess what happened? My name in the top one. You see that? So I'm proud of that. And in 2000, and I was invited by Nature Review Cardiology, asking me provide one comprehensive review about the diabetic cardiomyopathy mechanism, how it impact for the clinicals. So we provide 23-page review for this journal. How big is that journal? You know that. Nature is a impact factor 69, and the Science is 63. This review is 49. It's a very good journal, very solid. So we are happy. And only 2 years, already 120 citations. So more interesting, and you see this paper just published 2 months ago, right? And they can use some analysis. And if they ask, who is the first one to study diabetic cardiomyopathy? My name is there. And which institute? University of Louisville, right? And what is the labeling like? So you see, all this is like a feature that is kind of the disease like a marker, but only this one is the zinc supplementation. That is my marker. So now I want to come back to the story. In Toronto, I visit my friend, and after we talk, and he work on the FGF1 can replace insulin, because insulin can reduce the glucose, but sometimes reduce too much, make it hypo and glucose, right? Hypoglycemia, that cause some die. FGF21 reduce glucose like insulin, never go to normal level, go to lower normal level. And I work on zinc. You can guess what we see, why we are laugh. You see this? Dr. Bunting, Dr. Best, and they allow we are have the photos. And he worked on the FGF21. I work on zinc. And you see, that's why we laugh. What we wish in there, we don't know. You can guess. So I want to stop here and thanks my lab member and also is a collaborator and Dr. Tan and Jim Young. And Jim Young right now is faculty, is not my student, is a collaborator already. And Dr. Dan Conklin there. So we have a lot of collaborator in there, and Madhuri, and so we have nice. So I want to stop here and answer your question if you have. Do you have question online? Okay, good. Sure, then.
Thanks, Lou.
Thank you.
That was great. In that meta-analysis of clinical trials, were any of those looking at the polymorphisms? Because it seems like part of the problem with just giving zinc is all of the other factors that influence MT, which appears to be a really important.
For that, yeah, for that analysis, they won't do that detail. And even when I read the zinc analysis publication, actually there is not like an MT guy like myself. And MT really, if they have I know, even not diabetes, in the senior people, they have a low MT expression. So they have a zinc deficiency. They have some immunodeficiency. So that's why we are thinking about to collaborate with the paper first author published in same year and MT, and they are started working on the MT in the old senior people. So probably we try to collaborate with, but not like in the case yet. Thanks.
Just as a follow-up,
Sure.
were any of the clinical trials done with combinations of nutrients rather than just zinc? For instance, some NRF2 activators like sulforaphane, if they do that in combination, would you expect this would actually enhance?
Very good question. Actually, they have some several like a combination supplement, and why they try to combination, because even zinc is have like